So just be aware that we're not having class next week. So all of this stuff is going to be due 2 weeks from now. Let's see, that'll be the 5th, so that Monday after Easter. Great. There is the same format as the last couple of times where James put out a PowerPoint and his talk to the PowerPoint. We're going to go through some of that information today as well, just so you guys can get it from another perspective. Thanks, guys, for signing in. And then we're going to briefly touch on some of these. We're going to briefly touch on all of these topics that you're going to be introduced to today. And it is quite a bit, so just fair warning. Make sure you guys don't wait until, Sunday night, the day before it's due, to work on this stuff, because it is going to be quite a bit. You have your control of microbial growth. This is a Labster simulation. Pipetting, mastering the technique, also a Labster simulation. So there's 2 Labster simulations there. Gel electrophoresis, also a Labster simulation. So you guys have 3 simulations to do this week. Yeah, I could be getting those, or if you guys emailed me within the last day or so, I may not have responded yet. So I apologize if you guys haven't heard back from me yet. It looks like I do have some things in my inbox. So those, yep, yeah, no worries. And if you have questions, feel free to ask them in class here. So those 3 simulations, I think 2 of them are fairly short, but one of them is pretty long, like an hour long. So just be prepared for that. Microbial growth via UV radiation. There's an experiment that James talks through, so you guys are going to have to watch this video, read through the document, and we're going to touch on this topic a little bit, but we're not going to go into this document. And then there's also the Dropbox questions for this one. So I think there's only 1 Dropbox question. There's, of course, the quiz, and then there's 3 different simulations for this week. Well, to be done within the next 2 weeks. So let's get started here. I just wanted to, so I basically went through his PowerPoint and just pulled out some topics to talk with you guys about. And the ones that I wanted to hit on are things that people, have trouble with or have had issues understanding. So just wanted to hit on all those topics for you guys, hopefully in a quick overview, so that you guys can ask any questions that come up, or just are prepared for reading through those documents. So it's a pretty long PowerPoint. I think it's like 38 slides. Obviously, there's 2 different videos there that James is talking through it. But the first thing I wanted to talk about is the Spaulding classification system. So this is a system where we're talking about disinfection or decontamination. And if you think about like a healthcare setting, there's all sorts of reusable instruments that we use, such as, beds, bedpans, linens, thermometers, all sorts of things that are not necessarily disposable that we use over and over again. So there has to be some method of decontamination for cleaning. And so any institution is going to have a team that's focused on cleaning. So usually you think of janitors, but really there's an entire focused staff on these issues of cleaning and decontamination that's really necessary in an institutional setting. So the Spaulding classification is what we decide needs to be cleaned and how it needs to be cleaned. So I put here environmental sampling as well. The Spaulding classification really deals with stuff that's touching humans, whereas environmental sampling, this classification system really applies across a wide range of industries. So if you think of like food industry, like ice cream making or beer making or wine making, or development of pharmaceuticals, such as like contact lens solution. All of these things need to be done in clean environments or in environments where you're managing the microbial load on things. So they actually have similar classifications. Usually they use like a coloring system, like a red, green, yellow, like the stoplight system. And so the Spaulding system is similar to that. There's 3 different sets or stages. There's the critical, there's semi-critical, and there's non-critical. So first of all, critical would be any objects within the system that come in contact with normally sterile tissue. So this would be like anything you're going to be doing surgery with, anything you're going to be cutting bones, like a bone saw, anything you're going to be jabbing into people, like needles. So these are the things that need to be sterilized or decontaminated on a high level. So those things that are touching sterile tissues. Your semi-critical level is a little bit less than that. And usually if you're thinking of environmental, this would be like your red light. So things that are actually touching your food samples, those types of things. Semi-critical would be things that only make contact with your mucous membranes or the skin. So these are things that can be invasive, but they're usually only touching your skin or the inside of your mouth. So if you think of the dentist, they have to do a lot of decontamination because they reuse equipment as well. So think of like dental mirrors, picks, those kind of things. And then because those are all touching your mucous membranes. And then your non-critical items. So your non-critical items are those items that are usually only touching your skin or don't touch your mucous membranes. So these would be things like sheets on a bed or the bed itself or bedpans, things that are not normally invasive to your patients. So it's a really kind of a fun exercise to do this for your own house. To decide, you know, in your kitchen for your cleaning, for food preparation, which things are critical, semi-critical, or non-critical, which things are touching things that need to be sterile, which things are only making contact with those things, or which things are kind of secondary to that or not even touching it. To really decide what things need to be cleaned and what priority you should have for cleaning things. And this is an entire industry, industrial hygiene. It's a really cool industry, and people working in industrial hygiene all day long. So a really interesting field to get into if you're interested in this kind of stuff. So this next slide. So this is a slide I took from James's PowerPoint. It gets in a little bit more of the terminology that you guys need to understand. So we went over, I think we've gone over quite a few of these terms already. Aseptic, free of living pathogens. We've been talking about that since lab 1. Last lab 7, I think we talked about bacteriostatic versus bactericidal when we were talking about antibiotics. So again, acidal meaning to kill, static would be inhibiting or slowing down. Aseptic, antiseptic. So there's all sorts of terms to understand. So make sure that you guys understand the differences in these. And I kind of wanted to point out that there's kind of a level in different cleaning techniques. So I did smallest to the highest, with this highest one being the most, you know, aseptic or the most disinfected. So sterilization is really your, you know, number one. If things are going to be, you know, touching the blood, touching those sterile tissues, they need to be sterilized. And he goes into a couple of slides, so make sure you guys are reading that PowerPoint about different methods of sterilization. A couple of them that you can think of would be autoclaving something, so you're using heat and you're using pressure to kill everything, including the spores. You could also think of incineration is a method of sterilization. So when you are heating your bacterial loops, you're using extreme heat to basically burn off all of the microbes. And you can also use chemicals for sterilization. There's very specific types of chemicals you can use, and there's very specific protocols that you have to follow when you're doing sterilization to be ensuring that you're getting that aseptic sterilized surfaces that you're dealing with. And really what you use depends on what you're trying to sterilize. So you can sterilize wet things, but you need to do it without water. So you can sterilize dry things like metal trays and metal dental instruments, and you can use water for that. So there are different things that you can sterilize in different ways. For example, some of the chemicals are a gas. So instead of using heat for things that are sensitive to heat, you could potentially sterilize with a gas. And then moving down the chain from sterilization, there's high-level disinfection. And you guys can be following along in that PowerPoint if you want. You can find these slides. So there's a high level of disinfection, and this is going to kill most of your bacteria, and in most of your spores, but there may be some resistant spores still remaining. So a high level disinfection, I think an example of this would be pasteurization, where you're going to be killing most things, but you may still have some bacteria that was able to endure those high temperatures or pressures, and you may need to be checking for those contaminants, so those unwanted microbes. Your intermediate level is similar. You can use, I think, like bleach, alcohol would fall into your intermediate to low, where you're going to kill most microbes, but you're probably still going to have some spores or resistant microbes surviving. Then you could have your low level sanitization. So these are all usually some sort of chemical or some sort of method such as heat or pressure to be cleaning. So your low level sanitization might be like using soap for hand washing. So or cleaning in a washing machine, and using enzymes to break down fats and lipids. That's what your detergent in your washing machine does. So that would be considered like a low-level sanitization. And then at the base of all this is cleaning. And I don't want this to seem like this is the least important. This is probably the most important of all of these steps. So cleaning is that basic removal of dirt, or the disruption of that surface of dirt that's on things. So cleaning, again, would be like soap and water. And this is really the foundation of any additional level of sanitization. So if you're cleaning your counters because you've been preparing chicken, and chicken could be a source of salmonella poisoning, you're going to want to clean your counters with soap and water. Make sure that you've removed all of the large pieces of chicken, say, but then you're gonna next want to do some level of sanitization in order to kill those microbes. So there's those two levels, and cleaning really underpins all of the other levels. You're gonna want to wash your laboratory equipment before you autoclave it and it becomes sterilized. You're going to want to wash your, dental picks before you, sterilize them. So all of those types of things. So that really gets on to a new topic, sterilization. One of the methods of sterilization that I didn't touch on here is ionizing radiation. So you can use radiation to kill microbes and spores, and one of the ways of doing that is using ionizing radiation. So ionizing radiation, you can think of it, I think there was a cool little diagram that I wanted to show you guys that the CDC has here, which I thought explained it pretty well. So you can think of, radiation as all the wavelengths of light, where visible light is where we see here. And then the low energy radio waves, microwaves, infrared. Up here at the high end is your ionizing radiation. These are going to be your X-rays, your gamma rays, really high energy light waves. And then right at this border here is what we're going to actually talk about are the ultraviolet range of light. And so ionizing radiation will sterilize things, and that's those really short wavelength, really short wavelength rays. And then what we're going to be talking about is non-ionizing radiation, which includes UV rays, but it also includes microwaves, radio waves, light waves. But UV rays, especially in the range of 254 to 260, I think UV goes from like 4 to 400. But in this 254 to 260 range, you'd get damage to DNA. And when you damage DNA, obviously you're impacting the ability of microbes to replicate their DNA or humans to replicate their DNA, and you get things like skin cancer, or you get microbes that die. So the way that UV rays, there's 3 different types. There's UVA, B, and C, and I think it's the UVC that are in this range. Don't quote me on that. Again, that's in that PowerPoint. But these are the most damaging. To microbes and to humans. So this is really that type of UV ray that you're trying to avoid with sunscreen. And what UV does is actually when those particles hit your DNA, it actually causes the DNA to break apart and form what's called pyrimidine dimers. So again, you have your pyrimidine bases, and then you have your purine bases. So if you think of DNA as A, T, C, and G, your T and your C are your pyrimidine bases, and a pyrimidine dimer is usually a T to T linkage. So typically, again, your A, T, C, and G usually pairs with T, A, G, C, where T and A are usually linked. But when UV light hits your DNA, it causes 2 T's sometimes to pair with each other. And what it does is it causes a little kink in your DNA. And when your DNA replicating enzymes are going along, they'll actually fall off at this point because they can't make it over that little bump. So it inhibits the replication of DNA, and it causes issues for cells. So one of the ways that cells, that some bacteria or cells can get around UV ray or repair the damage that UV rays cause is called an SOS system. The SOS system is a system like an enzyme that'll actually go and attach to these pyrimidine dimers and cut them out of the DNA. So if this is your double-stranded DNA, it's happily going along, and suddenly there's a TET, and you get a little bump from the UV rays. This SOS system will come in and cut that section out of your DNA, remove it, and then fix that section so it's no longer a little bump. So it goes back to being flat. And this SOS system works pretty good, but the more UV damage that you get over time, so the longer that you're exposed to UV, the more and more of these happen in your DNA, and the less that this system can keep up with the damage. So eventually, if you get too much damage, you're gonna inhibit your DNA replicating. Whereas if there's, just low level, usually your body can keep up. Some bacteria don't have this system, and so some bacteria are really susceptible to UV decontamination or UV sterilization. But yeah, so this is the system that those bacteria that do resist UV might have this system. Other things that could resist UV would be your endospores. Your endospores will protect the DNA of the bacteria from UV. Also biofilms. So you create that mucus layer over top of the bacteria, and it protects them as a barrier from UV. So there's different things. So amount of time of exposure, the level of barriers that are existing, the wavelength of the UV, the different type of bacteria that you have different levels of susceptibility. So I think there's a slide that talks about the different impacts that UV has on cells, the different factors that go into how damaging UV is going to be. So make sure you look at those. But just know it's this SOS system that is repairing that UV damage. Yeah, I wanted to, because we were talking about DNA, I wanted to skip over pipetting for the time being and talk about restriction enzymes. So this is a whole nother topic. I think you guys are doing one of your labsters on this particular topic. But we just wanted to touch on restriction enzymes and what it looks like on a DNA fingerprint. Again, a lot of information and completely different topics, but bear with me. And if you have questions, let me know. So restriction enzymes, and I actually Googled it because I was like, wait, how would I show you guys what lists of restriction enzymes are? So this is just the Wikipedia page for G through K of the alphabet of the different restriction enzymes that exist. So restriction enzymes cut DNA at specific sequences. And so you can see here that there's like GST1 or GDI2. Usually a restriction enzyme is denoted by 3 letters and then a number, so HAE3 or HAE4, and it's indicative of the source of the restriction enzyme. So for HAC, it's referring to Halococcus actonifaciens. I'm not pronouncing that correctly. So usually the 3 letters refer to the bacteria that they found that restriction enzyme in, and then the number is which number of restriction enzymes they found. So apparently in this HAE, they found 4 different restriction enzymes. So what exactly is a restriction enzyme, now that you know that there's tons and tons and tons of them? So these are all the H's. There's so many restriction enzymes out in the world. So obviously it's a pretty important system if there's a lot of them. So restriction enzymes basically are what is used to cut DNA. Bacteria use them to protect themselves or to attack each other. They can cut, their rivals' DNA sequences, or they can cut up DNA in the environment and bring it into the cell, like we were just learning about last week as transformation. So restriction enzymes cut DNA, but they cut DNA, excuse me, coffee down the wrong tube. They cut DNA in a very specific way. So there's usually a specific sequence that they're looking for. So for example, people always use the example of EcoRI, and this is E. coli, so ECO is referring to E. coli, and then this is restriction enzyme 1 is what this is referring to. So EcoRI or EcoRI looks for this particular sequence and will cut the DNA between the G and the A. So I did some example DNA sequences here. So just think of these as, your normal DNA in your cell. If this EcoRI is introduced to your DNA, it will scan along and will look for this GA break, but in the context of this larger sequence. So it's going to look in both directions. And in this particular example, we have the GAATTC right here. So this, if we introduced EcoRI to this particular sequence, it would cut right there. For the second one, we see that it would cut right here. We have our GAATTC. We have a GA here, but it's not the right sequence. And then again here, GAATTC. So this is what it would cut as. So let's imagine we have an unknown restriction enzyme that cuts at all GA sequences. This may exist, but I don't know if it actually exists. So anytime there's a G and an A, it would cut between a G and an A. So for our top one, we would see that it would not only cut here, but if you scanned along here, there's another G and an A here. I don't see it anywhere else. For this one, there's a GA here. And for this one, there's a GA here. And I may, let's see, let's put in another GA somewhere here. Let's put an A here just for fun. So we have another cleavage here. Oh, so now we have 2 because it's actually GA in both directions. That'll be fun. Okay, so if we introduced a restriction enzyme that's going to cut DNA and we tell it to cut at G's and A's, which is what we just did, we end up with little tiny fragments of DNA. And these are what we refer to as DNA fingerprints. So if you're watching your, forensic files or Snapped or any of those other TV shows that exist where they're doing forensics, NCIS, all sorts of ones, they usually show pictures of these DNA fingerprints, and they're little lines, and then they compare the two, and they do paternity testing where you can see who's the father, or they do different things like that. So DNA fingerprints are really commonly used for identification of plant strains, of humans, all sorts of things. And there's actually other technologies as well, but restriction enzyme DNA fingerprints are one of them. So let's just say that these were our 3 different humans, and we were trying to see who was related to who. So for this top one, how we would do this is once we get the DNA cut up, we would run it on a gel agrophoresis. Electrophoresis. is what we would run, and it would look a little bit like this. So a gel electrophoresis machine is how we separate different lengths of DNA in a gel. So think of this gel as like a sieve where there's little particles, and usually what you're using in a gel is agarose or agar. So it's like a gel and there's little spaces in between. And if you have a really long piece of DNA, if you load your DNA up here, it's going to run this way, and we'll talk about why it's doing so. But it has to get through all of this gel that's in here. And so a long piece of DNA is going to get caught here, and then it's going to get caught here. So it's going to move pretty slowly. But if you think about like a short piece of DNA, like maybe this single G here, this short piece of DNA is just going to move right on through like that, like this gel doesn't even exist. So it's going to move really fast. So the further down, or the faster that the DNA is moving, the shorter that it is. So you have your long pieces of DNA up here, and you have short DNA down here. And you get basically a gradation of all of your different pieces as they go down this gradient. So why do they go down a gradient? So gel electrophoresis machines are charged. DNA holds a negative charge, is the charge on the DNA. And a gel electrophoresis has a negative end and a positive end. So because your DNA is negatively charged, it's going to be attracted to the positive end of your gel electrophoresis machine. So all of your DNA is going to move towards the positive end. So this is considered your anode, or your negative, and this would be your cathode or your positive. Hopefully I got that right. Sometimes I get that confused. So because DNA holds that negative charge, it's always going to run towards this positive end. And usually this positive end is considered red colored. And usually the negative end is black colored. So the little phrase that everybody says is that DNA runs to red. So it always goes towards the positive end, or DNA runs to red. So let's go back over to our little fragments that we cut up with our restriction enzyme, and let's see how we would deal with this. So first of all, you would have to count the number of base pairs that are in your DNA fragment. So for person 1 here, we have 1, 2, 3, 4, 5, 6, 7, 8 in this first segment before we get a cut. The next segment we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And our third segment we have 1. For this one we have 1. We have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. We have 2. We have 1. And we have 5. Because we have these little short segments here, because we had this A that we put in here. And our final one, we have 1. We have 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 1, 2, 3, 4, 5, 6, 7, 8. A 1, a 10, and an 8. So if we were going to load, so this will be, let's call them person number 1, person number 2, and person number 3. If we were going to load person number 1, so these are our loading wells in a gel electrophoresis. So the first well is usually not a sample. It's usually a what's it called? A not buffer. It's a ladder, a DNA ladder. And it basically is a standard that you can compare your lines to. So it's kind of your control. So our first sample would go in this line, and we see in our first sample we have an 8, we have a 10, and we have a 1. So the longer DNA is going to show up at the top, the shorter DNA is going to show up at the bottom. So number 1 here might look like something like this, where you would have a line for 10, we would have a line for 8, and way down at the bottom, the short DNA, we would have a line for 1. So that might be the fingerprint, 3 different lines because there's 3 different sections for our first person here. For our second person, we see that we also have a 1. So we would want a little line down here at 1. We also have a 10. So we would want a line up here at 10. So those lines should be even. We also have a 2. So we'd have a little shorter line because it's a little bit longer. And we have a 5. So this would be our DNA fingerprint for our second line. You guys may notice that there's actually, instead of 3 sections, which we had 3 lines here, our number 2 has 1, 2, 3, 4, 5 sections. So how come we only have 4 lines? And it's because 2 of our sections are the exact same length. So both of those 1 sections are going to land in the same spot, so there's only going to be 1 line. It may be a little bit thicker or darker if you were going to be looking at these on a gel, but there should only be 1 line. And then our last one here, you can see we have a 1. You can see that we have a 10. And you can see that we have an 8. So our last one, actually, if you guys are watching here, looks exactly like our first one. So if we were trying to figure out which 2 of these were related, we might say, you know, number 1 and number 3. And it's actually, they're the exact same sequence because they're just backwards from each other. So the DNA enzymes, or the restriction enzymes, read DNA both forward and backward. And so you'd end up with the exact same pattern. So number 2 was an interesting example. So number 2 is exactly the same sequence as number 3 with 2 different changes. So we changed in here, there's a CGGA, and in here there's a CGAT. So CGGT, CGAT. So there's a change right here. And then we also added this A in here. So there's 2 different changes is the only difference between number 2 and number 3. So this would be an example of a mutation. So if number 2 had a mutation and it changed from a G to an A, like it did here, you could end up with a completely different DNA banding pattern because of what restriction enzyme you're using, even though these 2 are very, very similar to each other. Just because we changed, so this one didn't actually change our pattern at all, but this one changed it by making 3 different fragments out of that 1 fragment. So when you get mutations in your DNA, you know, maybe caused by UV damage or maybe just developing antibiotic resistance can be done through mutation, just mismatching DNA. You can get completely new DNA fingerprints when you have mutations. Yeah, so hopefully this demonstrates a couple of things that people generally have a little trouble with. First of all is identifying how many fragments you have. So you would want to count all the different fragments that you end up with. The second one would be what happens when you have 2 fragments of the same length. So we have two 1-length fragments here, so it would just be one line instead of 5 different fragments that you would expect. You're only going to have 4 different fragments because all of your 1s are lining up. And then what happens when you have mutations? So mutations can cause giant shifts in these patterns of restriction enzymes that you get. And then this is how you DNA fingerprint something. So, imagine that this was, a paternity test or something like that. We might think that these two people might be related, or they might be the same strain of bacteria or something like that. And this is one way that you can trace diseases. So if you're thinking of, COVID-19 and it has different variants, so your COVID-19 is probably going to look like, 1 and 3. And then when you get a mutation, you get these different patterns showing up, or you get those changes in the DNA that you can track and say, this was our starting, we had a mutation, and then all of a sudden this is the pattern that you're seeing. So this is one way that you can track diseases as they move through populations as well. Very fascinating stuff. And this was just a blank electrophoresis gel that I did. Okay, so I could draw some of that on a different page, which we didn't do. So I'm going to go back now to our final topic, which was pipetting. So I know you guys have hit on this a little bit already, just throughout our various labs, but definitely wanted to focus on this a little bit. So this is the slide that's in James's PowerPoint. Definitely understand what these different points are on a pipetter. So pipetters, as you've been using in your simulation, are used to transfer liquid. You always want to use a pipette tip, which goes on the end here. So this F is the attachment point for your disposable pipette tip. E is your nose cone. So the nose cone is actually this white area here. So that's all E. The nose cone is the area where you need to maintain cleanliness or sterility, because this is the part that's going to actually be, potentially touching the jar that you're going to be putting stuff into. So if you imagine this is your tube and you're putting your pipetter into it, and that's your pipette tip, but above your pipette tip is that nose cone. So it could touch the edges of your tube. So you always want to be cleaning that area, or make sure it's staying clean. D is your digital volume indicator, and we're going to talk about that a little bit here, and they go into it quite a bit in the PowerPoint, so make sure you read through that. C is your volume adjustment dial. So you can't see it very well in this picture, but there's always a way to adjust the volume on pipetters. It's usually a dial here in this area, or you can turn the actual plunger button itself to adjust the volume. So C could be in various different places depending upon the pipetter you're using. B is your tip ejector button. So there's 2 different buttons. There's A and there's B. A is going to be what pulls up and pushes out liquid. So that's your plunger button. B is what removes the pipette tip. So if you accidentally push B when you mean to push A, you're going to lose your sample and you're going to lose your pipette tip and you're going to make a big mess. But if you press A, you're not going to be able to change pipette tips between samples. So they're both very important. A would be your plunger button, and B would be the button you push to eject. And A actually has 2 different stops. So there's a, so if this is your plunger button on your pipetter, it goes down to a first stop, and then it will go all the way down to a second stop. So you can push it through both of these, but basically the first stop is what you push it down to do your accurate measurement. I should go over and then I can actually write that. So the first stop is for the actual volume that you're pipetting. The second stop is to remove all the liquid from the tip. So it actually pushes extra air in that distance. It pushes extra air through the pipette tip, and it is able to remove, you know, every last drop of liquid that's in your pipette tip. So in order to pick up liquid, you would press the plunger button to the first stop. You would put it in your liquid. You would pull it up, and it would pull up that amount of liquid. You would transfer it to your container. You would press through the second stop to eject all of the liquid from your pipetter. Just a random note there. And I think you guys are going to be doing that in the simulation, so you can understand that. So let's talk about this volume adjustment dial. So each of these pipetters is going to be for a different volume. Let's reset this to a reasonable level. So there's different levels. There's, let's say like a P10, a P20, a P200, or a P1000. So each of the P's is referring to a pipetter. And these are referring to microliter measurements. So a P10 goes up to 10 microliters. A P20 goes up to 20 microliters. A P200 goes up to 200 microliters. And a P1000 would go up to 1000 microliters or 1 milliliter, which is what this is showing right here. It's showing that 1 milliliter is equal to 1000 microliters. So being able to do the conversion between milliliters and microliters is very important. So the way that you would do it is whatever your milliliter number is, you would move the decimal point 3 places to get microliters. Opposite that, whatever your microliter number is, you would move it to the left 3 places to get your milliliters. So if you had 1000 microliters, you would move it to the left 3 and you would have 1 milliliter. If you had 1000 milliliters, you would move it to the right 3 and you would actually end up with 1 million microliters. Micro with little u liters, because you'd move it to the right versus moving it to the left. So microliters to milliliters, 3 decimal places to the left, or basically dividing by 1000 milliliters to microliters, 3 to the right, or multiplying by 1000. Anyway, so make sure that you guys are able to do that. So when you're looking, whatever pipetter you have, for example, this one could be a P10. This is the maximum amount that you should be pipetting with that pipetter. So if you wanted to pipette out 15 microliters, you should not use a P10. If you wanted to pipette out 15 microliters, you would probably be using a P20. So there's actually a low and a high. I think the P10 goes from like 2 microliters, don't quote me on that, to 10 or 1 to 10. This one might go, this one's a 2 to 20. This would be a 20 to 200. And this one could either be a 100 to 1000 or a, depending upon the pipetter, probably a 100 to 1,000. So this is the low end you should be pipetting, and this is the high end. So actually, this one's probably 0.1. So it's usually like an order of magnitude difference between the low and the high. So if we wanted to measure out on a P10, let's say we wanted to measure 9 microliters, how would that look on the volume adjustment dial? So the way that these are set up is there's 3 different units. In some cases there's 4, and we'll touch on that. But typically there's 3 different units. The top one is going to be the highest decimal place that you can go. So in a 10, the highest decimal place would be your tens. This would be your ones. And this would be your 0.1s. For a 20, 20 is also tens, because that is a 10 decimal point. This would be ones, and this would be your 0.1s. For a P200, though, because 200 has a 100 decimal place, this would be your hundreds. This would be your tens, and this would be your ones. And then for a P1000, the highest decimal point is your thousands. So this would be, your 1,000. This would be your 100. And this would be your tens. So depending upon the type of pipetter that you have, that each of these boxes indicates a different level. So let's say we wanted to measure 9 microliters. How would that look on here? So 9 microliters doesn't have any tens, so this would be a 0. How many ones does it have? It has 9 of them. And how many 0.1s? It has 0. So on a volume adjustment dial, you would change it to 090 for 9 microliters on a P10. What if we wanted to measure 9 microliters on a P20? There we go. Sorry about that. Just opening all my sticky notes. Yeah, so this would look exactly the same on these two. So what if we wanted to do, instead of 9, what if we wanted to do 20 on a P20, 20 microliters? Our tens, we would have 2, because it would be for 20, so you'd have 2 tens. You'd have 0 ones, and you'd have 0.1s. So your P20 for 20 microliters would say 200. So you might think that looks like 200, but it's actually 20, because your top is your tens. Let's go over to the P200. What if we wanted to measure, let's go 174 microliters on our P200. What would that look like? So 174, how many hundreds we would have? We would have 1. How many tens? 7. How many are tens and how many ones? We would have 4. So this is what 174 microliters would look like on our P200. You'll notice that our P1000 could also measure 174 microliters, because it's within this range of 100 to 1000. So what would 174 look like on our P1000? There is no thousands, so there would be a 0 there. There's a 100 there for that 1, and there is 7.4 tens. So usually your last place can go to a decimal. So you could either write this, or you could write it as 017, and I would probably accept that too. So I mentioned before that sometimes there is a 4th box. So instead of just the 3 boxes, like here, oops. Sometimes there's a 4th box. So if we, add another box here. on the end. And this would work the same way. You would have 1000s, your hundreds, your tens, but you would also have a ones spot. So 174 on a 4-box P1000 would be 0, 1, 7, 4. So always your top box is going to be your highest value that you could go to, or the highest decimal place. So make sure that you guys practice those. There's a bunch of practice examples of that in the PowerPoint, as well as in the document down here. So make sure that you guys understand how to do the pipetting. So those are all the topics I wanted to hit on today. It's actually